Concept 26.5 says molecular clocks help track evolutionary time. We've already talked a little bit about the fossil record and how geologic evidence can help pinpoint a certain time frame. We know the process for laying down sediment and forming that sediment into rock, and so based on the strata and the layers we can make time frame estimates. Then of course we can do radiometric dating that also helps pinpoint time, but there's also a molecular way, a genetic way to take a look at time frames as well, and those are using what we call molecular clocks. One of the long-term goals of evolutionary biology is to understand the relationship among all organisms all the way back to the very beginning, and that would include creatures for which there is no fossil record, whether they are soft-bodied or perhaps they didn't leave fossils or we haven't found those fossils, or perhaps we are reverting back to a time frame billions of years ago to where we have very few available fossils from that time. So when we extend molecular phylogenies beyond the fossil record, we can no longer rely on some of the other varieties of evidence. We have to rely on a few assumptions, important assumptions about how change occurs at the molecular level if we want to try to develop some sort of time frame or clock. Now you want to note that these assumptions are evidence-based and they're always open to revision with further experimentation and data. So yes, these are scientific assumptions that we are having to make, but they're not wild guesses. They're based on all the available evidence, and from there we can extrapolate out to get to where we need to go. The process for doing this is called the use of a molecular clock. A molecular clock uses the constant rates of evolution in some particular genes to estimate the absolute time or the actual year, date range time of evolutionary change. In orthologous genes, nucleotide substitutions are proportional to the time since they last shared a common ancestor. So if we're looking at the orthologous genes that two species share, the number of changes or nucleotide substitutions are going to be proportional to the time going back to when they were present in the common ancestor. In paralogous genes, or paralogous? Let's go with paralogous. In paralogous genes, nucleotide substitutions are proportional to the time since the genes became duplicated. So we have a proportion of time between the various mutations and changes that occur in the genes and the time from when those genes came into being, basically from the point at which they were duplicated or from the point at which they split off from their common ancestor. So the greater amount of time that has passed, the greater the observed genetic change in these particular genes. So it's a comparative tool to see the more changes that are present. That generally means the longer the time period that has gone by. A good question would be, how do you set these clocks? How do you know what that time frame is? Well, molecular clocks are calibrated, I guess you could say, against branches whose dates are known from the fossil record. So the way that we set these or calibrate these molecular clocks is by comparing them to other data that we have. So we would go, say, look at the molecular changes that have occurred in a particular set of genes over time, and we would compare that to the fossil record that we have for those same species. And so this would be very similar to the way you might calibrate a scale with a known mass. If you want your scale to read 1 kilogram and you have a known 1 kilogram mass, you can calibrate that scale. Or if you want to zero out the scale to calibrate it, then you can feel stronger about that scale's accuracy. When scientists use these molecular clocks, they try to avoid using only a single version of this, a single gene, because individual genes can vary in how clock-like they are. This is well understood by scientists that there are variations within the rate of change between different genes, and so you're not going to get the same exact number every time using this method. An example of a molecular clock is shown here in your textbook. It's a molecular clock for mammals. It basically shows the number of accumulated mutations in 7 particular proteins, how that has changed over time, and how those mutations have accumulated, have increased over time in a fairly consistent manner with most mammal species. So if you look at a divergence time between 2 particular species, and if you stretch that out from the point of divergence where they had the common ancestor to 30, 60, 90 millions of years, you see the number of mutations jumps along with that. And so for the most part, you can draw a best fit line and you see that it is fairly fairly accurate, fairly linear in most cases. Now you do see a few outliers. For example, you see these green dots that are a bit of an outlier. These are primate species, and their proteins appear to have evolved more slowly than most other mammals. They are outliers. But if you look at the majority, you see they follow the same trend if you draw a best fit line in there. So it's well known amongst scientists that these molecular clocks are not perfect, that you have to use a variety of tools if you want to feel confident about your data, and this is the case with something like a molecular clock as well. There is an idea in this thought process of using molecular clocks to study phylogenetics, this idea called the neutral theory. The neutral theory states that a whole bunch of the evolutionary change that occurs in genes isn't helpful or harmful at all. It's simply neutral. It doesn't really do anything. It has no effect on the fitness of the creature, and so if it has no effect on the fitness of the creature, it's not going to be influenced by natural selection. So it states that the rate of molecular change in these genes and proteins should be regular, like a clock. So if the changes that occur don't negatively or positively affect the creature, you can expect that natural selection is not going to influence them, and that they should be basically randomly regular, if you could think of that as a particular way of saying it. The problem is that in many cases this is not reality, that a lot of changes in the gene code, a lot of changes in the genetic code or the proteins of the creature, they are either positive or negative, and so they are affected by natural selection. Differences in the clock rate for different genes are really a function of how important that gene is. If that gene is particularly important, if the exact sequence of amino acids that a gene specifies is essential to survival, most of the mutational changes will be harmful and only a few will be neutral. And as a result, such genes change only very slowly. Like we were talking about the genes that code for the ribosomal RNA, they change extremely slowly because for the most part changes are detrimental and get weeded out, don't get passed along. But if the exact sequence of amino acids is less critical, fewer of the new mutations will be harmful and more will be neutral, and those genes will change pretty quickly, pretty rapidly. So if you have a lot of neutral changes, you could expect that not to be affected by natural selection, keeping it down. And the opposite is true. If those changes are positive, you would expect natural selection to increase that likelihood within the population. So this neutral theory states that if you can take away natural selection, and if the changes are not helpful or harmful, they should occur at a regular rate. And that does seem to be the case, but the problem is in many instances the changes are affected by natural selection. So the problems with molecular clocks include the fact that the molecular clock often doesn't run as smoothly as the neutral theory predicts, because small irregularities can either have a positive or negative effect, and then they are going to be affected by natural selection. Some of those DNA changes are going to be favored more than others, so that would affect basically your run of your clock if it's speeding up or slowing down. Estimates of evolutionary divergence older than the fossil record have a higher degree of uncertainty, and this makes sense. If you're relying only on molecular clocks for your data and you don't have any other source of data to kind of check that or balance that or calibrate that, then you're probably going to have a higher error rate. We have fossils dating back to over 3 billion years ago, but those are very rare and they're not very common. Most of our fossils, the abundance of fossils we have, takes us back only about 550 million years. So once you go beyond 550 million years, you have less other outside resources to calibrate your molecular clock with. And the use of multiple genes to calibrate molecular clocks is a better idea, because that helps improve your estimates and even out the effects of natural selection. And these are all well-known issues in biology, and this use of molecular clocks is something that's discussed or still debated within biology and amongst scientists as to how effective they are and what's the best way to tweak them to get them to be most accurate. It's a continuing source of debate and investigation, which is good. It makes our science better, and it makes the information that we have stronger. Your textbook gives a great example of applying this molecular clock to a particular instance, like for example the origin of HIV, the virus that causes AIDS. Phylogenetic analysis shows that HIV is descended from a virus that infects chimpanzees and other primates. What's weird is within the original primates, the original native hosts, they don't get the AIDS-like disease. So this virus is one that mutates very often, and it's very much affected by natural selection, which most viruses are. They tend to mutate and change quite a bit. And in its jump from other primates into humans, the virus mutated and changed. And in humans, it does oftentimes go on to cause the AIDS-like disease. Based on genetic analysis, HIV spread to humans more than once. There's actually multiple variations of the virus within the human population, which makes it likely that humans were infected in multiple separate cases or instances. Comparison of HIV samples show that the virus evolved in a very clock-like way. The virus has a tendency to change in a pattern that can be looked at and noted over time. Application of a molecular clock to one strain of HIV suggests that the strain spread to humans during the 1930s, which is kind of fascinating to think that this outbreak of this disease began long before I was alive, or of course you guys were alive, and before this was any part of the news. It wasn't until probably the 70s or into the 80s that this really became noteworthy and newsworthy. But this disease has infected the human population for quite a bit longer than that. Of course, it came to prominence in the 80s when it became an outbreak and began to spread quite a bit more. So here is a good graph that kind of shows what they're talking about. This is figure 26.2. The black dots you see here are data points based on the DNA sequence and the changes of the DNA sequence over time. You'll see we have this information from the mid-80s on up and through to the 2000s, and looking at the rate of change in that time frame, if we extrapolate backwards, basically you can see the red line, and then you can see the range. Of course, it widens as you go down because you have to have some measure of not being sure, of course, because you are extrapolating. So you got an adjusted best fit line, and then you take all that all the way back, and it would seem to indicate that the first infection of humans occurred sometime between 1920 and 1940. So 1930s, somewhere in the 1930s would be your best bet. So that's a good example of a molecular clock in action. A great example of using it for kind of a modern day issue that we're looking at, but of course we also use it when we're doing these phylogenetic studies of species living millions of years ago. Alright, let's take one last break and then we'll come back for concept 26.6, wrap it up.